Okay, good afternoon, and thanks to the organizers for the opportunity to present here. I'm going to talk about connecting molecules and organizations, and it's probably more linked to the previous session about distributed systems, but anyway. Starting point is INTACT. INTACT is a curated molecular interaction database. It holds currently 1.2 million binary interactions as of the release last week. About 50% of these are from direct author submissions. The other 50% are from curator-based choice based on funding or curator interest or expertise. And we have, from the very beginning of INTACT, more than 20 years ago, always focused on a detailed deep curation model. So not only A interacts with B, but what are the experimental technologies, which have been used to determine the interaction, what are, for example, the interacting domains if it's in the data, how have the proteins been modified in the experimental context to be able to later on do judgments like what is actually the probability that this is really a truly happening interaction and not an experimental artifact. And that means that you can then in the display, for example, highlight, show me all the interactions which involve on one or both sides of the interaction variant of the canonical protein as opposed to just the standard uniprot protein, like what is highlighted here in pinkish color. You can also select what is displayed in the INTACT interface based on multiple criteria like the confidence score. You can then get even more detail if you click on the tabular representation on this sort of magnifying glass, and then you get into the detailed viewer where you see individual protein domains, where you see tags which have been added to the protein, and you see, as I mentioned earlier, potentially interacting domains linking the two proteins if that data is available. If you want to go into even more detail, then you can use the Cytoscape app, which allows you to explore all of this interactively, and you can choose which features are displayed in the interactive Cytoscape display. And of course, you can then interact with all the other Cytoscape features. INTACT is not the only interaction database. There were already early on, there were a number of interaction databases around, and we got together in around 2002 with the first ideas and the first concrete activities in 2004 in the International Molecular Exchange Consortium. And this has had over time 12 international contributors from 4 continents. Not all of them are active anymore. And we've heard this in a couple of other slides earlier. Part of the agreement between IMEx partners is to stand in for each other. And it has happened multiple times that one of the partners became inactive because of loss of funding or whatever. And all of this data has not only been saved on an FTP server, but it's actually now part of the IMEx data set still. And we are still maintaining the mappings. We are updating uniprot mappings for the proteins. So the data is really not only saved, but maintained, because from the beginning, we focused on setting up ways where we can do this maintenance job. And it developed really nicely from common file formats in 2004, where we just agreed, okay, let's all provide our data in the same format so users have an easy time to get it together and to select all the data they need for their analysis, to then common curation guidelines. Clearly, when you want to combine data, it's not only important that it fits together syntactically, but also semantically. And so we agreed how we do the curation of the data. And then we developed a lot of tools over a series of grants. It was also quite a nice sort of handover from EU funding in the first CIMEx grant to UK BBSSC and NIH, NIGMS, IMEx funding, and we have achieved for Intact and MINT as representatives, ELIXIR core resource recognition, but it was really over all these years nice to see and to be built on the trust between the resources, increasingly more integration from file formats and common curation guidelines to now all active IMEx partners use one web-based curation platform, so we don't have 10 different systems which ensure the data input, but only one, which of course makes it more harmonious, but importantly, the expensive part. It's more expensive to have a writing system, because if you mess that up, then the results can be drastic. Than if you have just your reading web display system. But we have only one curation system, which we all share. And so a lot of the redundant work is taken out. And partners can focus on what they provide best, their expertise in, for example, a specific domain of interactions. And this has all been documented in publications over the years. One interesting example is MatrixDB, which I've selected here because they are based here in Lyon. And MatrixDB under Sylvie Ricard-Blum is a very small operation. It's basically Sylvie and various students over the years who have helped to develop the system, but they have expertise in the curation of the extracellular metrics. And so they can focus on that rather than having to maintain a lot of technical overhead for the curation system. And they then they contribute to the overall set of data, which is provided by through the Intact website. All the IMEx data is provided through the Intact website. So Intact benefits, but also they benefit because they have the editing tool and they have their own website, which allows to focus on the specifics of this extracellular metrics domain. Curation principles we've built on from the beginning is, as I mentioned, detailed curation. We have, for example, a very comprehensive ontology for controlled vocabulary for experimental methods. Objectivity, we want to make sure we don't make judgment calls when we do the curation. We want to represent the paper content and then carefully annotate methods to then allow these judgment calls to be done on the aggregated data. We always want to have the underlying evidence. We have always double checking between junior and senior curator. And all of this is codified in an externally accessible curation manual, so everybody can look up how we are actually doing these things. And one thing this allows is confidence scoring, which is not based on machine learning, like most published confidence scoring systems, but it's a heuristic system, which is based basically on a number of independent reports of an interaction, the experimental technologies which were used, and this is then scored and aggregated into a score between 0 and 1, and has shown usefulness, for example, in analyzing a large-scale experiment where the last third of the data essentially was randomized due to a mistake in the submission, and these randomized interactions got a much lower MI score than the proper ones which were in the first part of the publication. All of this is accessible through the website and can be used to retrieve interactions according to different cutoffs. The key part is that our detailed annotation allows us to do filtering to different criteria. And one example is at some point we did an almost full curation of all interactions from the literature around the LARC2 protein. And the left side here shows the more than 1,000 reported interactors of LARC2. which of course is a bit unrealistic, but this is what we took from the literature. And then we filtered. And the right side here shows those interactions which were determined under at least near physiological conditions. And you see it's only a handful. The smaller one is human, the larger one is mouse. And so if you take one thing away from this, it's be careful about what were the experimental conditions for data reported in the literature. How realistic were these conditions? And so that was really the key message we had in this publication. The detailed annotation also allows us to focus on variance, and we have a key data set which is continuously being maintained, where we annotate the effect of sequence variation on the interaction, whether the interaction remains the same, whether the interaction strength is increased or decreased. And this is now, this comprises more than 72,000 individual interactions, effect annotations. It's now part of the data for PEPWeb, the variant effect predictor, which is used in Ensemble. And it was only possible through the joint work from 10 IMEx partners over more than 10 years. Another key data set was the COVID-19 data set, again, because we quickly agreed on what to do and what together we could fairly quickly come up with a large set of COVID-19 related interactions, which was then published and maintained over the COVID period. And is still maintained in the sense that we still do the appropriate identifier mappings, but of course we're not focusing on this anymore. Now, slight change of gears for a moment to Reactome. Reactome is the, to our knowledge, largest database of curated human pathways, turning textual representations like these into user-friendly graphical representations and detailed molecular maps. And it's same as Intact IMEx. It's an ELIXIR core resource. It's also a global core biodata resource. And I'm not going to talk in detail about Reactome, but one thing we provide is in the detailed molecular maps, you can see for each protein a little red bubble in the corner, and that indicates the number of high and medium confidence interactions from Intact. So then you can click, and other IMEx partners, and so you can click on this and get a sort of halo around the protein, see the other interactors. You also see the structures if the structural data is there, which of course now is practically for everything with AlphaFold. And you can then explore this data further. You can also, it's integrated into the Reactome search, so you benefit from all the features of integration and a large increase in the coverage of Reactome, because if you assume there's around 20,000 human protein coding genes, 11,000 are annotated in Reactome. For around 7,000, depending on the definition, there's simply no functional annotation, so it's difficult to integrate it into a mechanistic database like Reactome. But the coverage in Reactome is increased by about 5,000 interactors due to the integration of these interactors from Intact. So this was just to show the reuse of the data and the integration across databases. Now, a quick excursion, because it was, and I changed this and put it in after the discussion around Civic, and this is credit where credit is due. Reactome had 800 contributors since 2002. Less than 30 of these were paid curators. The paid curators probably have done the lion's share of the work, but many of the individuals have done a lot of small and often larger contributions. We can only pay these volunteers by giving credit. And we've always provided DOIs to make pathways citable, but we've also always recorded the names and later on the ORCID IDs of contributors. And a while ago, we have changed Reactome, so you can search with your own name, and you get the contributions of that person to Reactome across pathways, reactions, etc. And then you can just, if you are that person, you can click, you get into the ORCID interface, you authenticate yourself, and then you can claim all your contributions to your ORCID profile, and you see then pathway curation in the same way in which you see, let's say, your bioarchive papers or whatever as your contributions to the scientific record. And that is part of our effort to value more than just publications as valuable scientific output, to make it citable, to make it traceable, and to make it really visible, as I have done a lot of curation work, whether I'm a volunteer or whether I'm a professional curator. And this is, I think, a big part of making data first-class citizens in scientific output. And this was already published a couple of years ago. There's now an active ELIXIR implementation study where we are aiming together with other partners on the study to extend this approach to data from INTECT, biomodels, PFAM, and the idea is to propagate curation contributions from these resources, both to ORCID profiles, but also to Epicuron, which is a specialized resource which aims to collect and aggregate curation contributions to the community. And there you have things like medals, leaderboards, and so on to give credit. And so we hope, again, that this is a step to really reward and give credit for something which is not just I have published X and Y. And with that, I haven't talked about machine learning, but I would like to make a short shout out. Bridge to AI was already mentioned in one of the early talks by Tom in the first session. It's an NIH-funded project to generate flagship data sets and best practices for artificial intelligence and machine learning. And the key part probably here to mention is it has also a training component. It provides full year-long lecture courses which give you a chance to brush up on AI technologies if you or your students so wish, and it's linked also via the QR code there. And with that, I'd like to acknowledge the INTECT and Reactome teams, the many contributors to IMEx, Uniprot, from Uniprot, collaboration with EuroPMC, and many others which have made all of this possible. And of course, the funders, US NIH, EU, UK BBSRC, and EMBL core funding. Thank you.